This is a short video overview of the neuronal cell recipe that can be found in our recipe store. This image is of a neural cell, and what we're looking to do here is to segment the nucleus, the cell body, and the axons. So this recipe is broken down into 4 chapters, where the first chapter looks to identify the entire cell. The second chapter identifies the cell body. Third chapter finds the nucleus, and the last chapter finds any of the axons protruding from the cell body. So the recipe itself is fairly simple, and what we're able to do is, first thing is just by identifying the green channel, which just gives us the general, I believe, the cytoplasm stain, or maybe the cell membrane stain. We're able to pick up the entirety of this cell just by using simple basic thresholding, dilation, and general manipulation of this segmentation to give us a clean, reliable result. Next thing we do is we actually identify the cell body by finding where the red channel is, the red protein localization is, segmenting this and then manipulating this using our dilation and segmentation to give us the results that we're interested in. So the one thing that we did here is we found out that the only thing missing from our dilation of the cell body was actually just the nucleus. So we're able to find the nucleus using the blue channel, do a segmentation, and then recombine this with our cell body chapter, or the neuron body chapter. From here, we are able to clean this up and give us a rough selection of the cell body. Lastly, if we bring back our mask that contains our whole cell, we're able to subtract the cell body to result with just the axons. What we're able to do is generate measurements like axon branching and axon thickness, which gives us a good idea of the morphology of this cell. Another thing that we might be interested in is the localization of this red protein, and we can do this very simply by segmenting the red protein and then telling us, basically doing simple binomial calculation between the cell body and the axons to give us the area fraction measurements of in the location of this red protein, or the red marker. It's a fairly classic example, and the kind of the approach that we would take towards colocalization studies. If you need help optimizing your own recipe for your own set of images for any sort of fluorescence, colocalization, or cell morphology studies, feel free to reach out to our application staff for input and assistance. Thank you.